Let's begin with the sympathetic system first. So the preganglionic neurons of the sympathetic system originate from the thoracolumbar segments of the spinal cord, specifically the lateral horn, and I'll show where that is coming up, or the intermediolateral cell column, so the lateral aspects of the spinal cord, if we were to look at a cross-section of the spinal cord. And then they are going to, so here's that segment of the spinal cord, thoracolumbar T1 to L2. Then they are going to have a short preganglionic neuron that will travel a short distance out from the spinal cord itself and then synapse onto a ganglia. So that can be a ganglia that is a part of the sympathetic chain as option 1. A ganglia that is not a part of the sympathetic chain, we refer to that as a collateral ganglia. That will be option 2, and that will most likely go to the gastrointestinal organs and tract. And then the third option will be the adrenal medulla, and we'll talk about these more specifically. But these are the 3 options for synapsing of a preganglionic neuron arising from the thoracolumbar segment of the spinal cord. From there, there's going to be a long postganglionic neuron, all right? So a much longer postganglionic neuron traveling from that autonomic ganglia all the way out to the effector organ that it is innervating, the effector organ in question. So that can be in the lacrimal glands, salivary glands, in the GI tract, genitalia, etc. So the difference here is that because these ganglia are so close anatomically to the spinal cord, there's going to be a short preganglionic neuron and then a longer postganglionic neuron. All right. Okay. So here is the third option, right? The options for synapsing of that sympathetic preganglionic neuron that exists outside of the sympathetic chain. So we said the sympathetic chain was one option for that preganglionic neuron to synapse. The second option we said was a collateral ganglia that was outside of that chain. And then the third option was the adrenal medulla. Now the adrenal gland, just to kind of zoom out here and talk about where this gland is, these are 2 endocrine glands that are located above the kidney, so they're bilateral, and they are basically divided into 2 different parts. There's the adrenal cortex, which is going to be involved in cortisol release and aldosterone release. We'll talk about that when we look at the endocrine system. And then there is the medulla, which is the inner darker staining part of the gland, and that's going to have cells that are called chromaffin cells. All right, so the chromaffin cells here are going to be the third option to which these preganglionic neurons can synapse. And then essentially the chromaffin cells here can act just like postganglionic neurons. In fact, scientists took a bunch of chromaffin cells and they placed them into the correct medium, and these chromaffin cells actually began to grow axons. So these chromaffin cells act just like postganglionic neurons. They have the same neurological origin. So in terms of the development of the chromaffin cells, they are derived from nervous tissue or neural tissue. And so they're going to behave just like a postganglionic neuron. They're sometimes referred to as a modified postganglionic neuron. And then they are going to also release their hormones, which can be epinephrine, about 80%, norepinephrine, about 20%, and then a very small amount of dopamine. And so the way in which that these chromaffin cells release this hormone is a very similar way in which postganglionic neurons synapse onto effector organs or effector glands. All right. So this is an example of the third route that a preganglionic neuron can take to result in effector organs being innervated and then having that autonomic response on these effector organs. All right. So here is a better schematic of that. So here's our spinal cord thoracolumbar segment, so T1 to L2. Here is the lateral horn. So when we talk about a cross section of the spinal cord, we've got a ventral horn, we've got a dorsal horn, and then we've got the lateral horn, as well as the intermediolateral cell columns, which is somewhat the same anatomical region of the spinal cord. So this is the specific location where the cell bodies of these sympathetic neurons are located and where these neurons will originate. They're going to travel out from the spinal cord. They're going to synapse, again, 3 options for this preganglionic neuron to synapse. It can synapse 1 onto the sympathetic chain. Another way to say that is a ganglia that is a part of the sympathetic chain, right? An autonomic ganglia that is a part of this chain. The second option is an autonomic ganglia that is not a part of the sympathetic chain, referred to as the collateral ganglia. Again, mostly innervating the gastrointestinal organs. And then the third option for communication would be the chromaffin cell of the adrenal medulla. Okay. Either way, irregardless of where these preganglionic neurons synapse, the resulting action will be that the postganglionic neuron or the chromaffin cell, which is a modified postganglionic neuron, will then release its neurotransmitter, and that will result in an effect on the effector organ in question. All right, so that's just kind of going over the way that the sympathetic nervous system is organized and the way that that can communicate. Now, if we look a little bit more closely at the anatomy of the sympathetic nerves, so if we look a little bit closer at the preganglionic sympathetic nerve, so they're arising from the lateral horn, they're traveling out through the ventral root, right? So the ventral root coming out here. And then they're going to enter the spinal nerve. And then they're actually going to synapse. They're going to leave the spinal nerve, synapse onto this ganglia. So here's the autonomic ganglia, where the preganglionic neuron, which is the lighter blue color, can synapse onto the postganglionic neuron, which is the darker blue structure. So the preganglionic neuron synapsing onto the postganglionic neuron, and then that postganglionic neuron will re-enter the spinal nerve, and then it will travel out to the effector organ in question. And if you look at the myelination of these 2 neurons, we're going to refer to the preganglionic neuron as being the white ramus because it is unmyelinated. And then we're going to refer to the postganglionic neuron as being gray ramus because it is, or sorry, the white ramus was myelinated and the gray ramus is going to be unmyelinated. Okay, so just looking at the anatomical makeup of these neurons as they enter the spinal nerve and then synapse onto the ganglia and then re-emerge to enter the spinal nerve again. The preganglionic neuron is myelinated, referred to as white ramus or white rami fibers. And then when it re-emerges to re-enter the spinal nerve as the postganglionic neuron, here we're going to refer to it as being a gray ramus because it is no longer myelinated. It is going to re-enter that autonomic spinal nerve. Okay, and so this is another example of where we talked about autonomic neurons being unmyelinated because that was somewhat unnecessary in the autonomic nervous system. So when we think about the somatic nervous system, those neurons need to be heavily myelinated because those signals need to be faster. They need to be more responsive, more immediate, right? When I move my skeletal muscle, that needs to happen automatically. Or that needs to be more responsive, more immediate, right? Happen faster. In the autonomic nervous system, these neurons don't need to be myelinated. They're smaller neurons, and they don't need to be as responsive as skeletal muscle, motor neurons do. Okay, so again, just reiterating, the postganglionic neuron will leave the ganglia as a gray ramus fiber. It will re-enter the spinal nerve here, and then it's going to then travel in the spinal nerve all the way out to its effector organ. And then we also have the other options being the collateral ganglia as well. Okay, I'm going to pause and see if there are any. So we said sympathetic nervous system, T1 to L2, a short preganglionic neuron, a long postganglionic neuron, and then we also described 3 options for that preganglionic neuron to synapse, either onto the sympathetic chain, or onto a collateral ganglia, or onto the adrenal medulla chromaffin cells, okay? The third thing that we said here regarding the sympathetic nervous system is that we have a myelinated white ramus fiber emerging from the ventral root of the spinal cord, joining the spinal nerve, and then we have it synapsing at the autonomic ganglia that's a part of the sympathetic chain. Then we have a gray ramus fiber emerging from that ganglia, re-entering the spinal nerve, and then going all the way out to the effector organ. All right. Let's switch gears and look at the parasympathetic nervous system and talk about ways that this differs. So the preganglionic neurons in the parasympathetic nervous system arise from the brainstem and the sacral spinal cord. So the brainstem here, we have these preganglionic neurons, and notice that the preganglionic neurons here are longer. So this is reversed from what we saw with the sympathetic nervous system. There's a long preganglionic neuron synapsing onto a peripheral ganglia that's way far from the spinal cord or brainstem. So as a result, this is going to have to be a longer structure to go all the way out to this ganglia, which is much further from the brain or spinal cord, much further from the central nervous system. And then from there, we have a short postganglionic neuron that's going a short distance from that parasympathetic autonomic ganglia out to the effector organ in question. All right, and that's going to be true for the brainstem, where we have the cranial nerves 3, 7, 9, and 10, as well as the sacral spinal cord, where we have S2, S3, S4, giving rise to the pelvic nerves. So the pelvic nerves will do the same thing. There's going to be a preganglionic neuron traveling a long distance from the spinal cord, the sacral spinal cord, all the way out to a peripheral ganglia that is close to the effector organ. And then we have a postganglionic neuron, which is much shorter in length because the distance between the peripheral ganglia and the effector organ is much shorter, and then that will innervate the effector organs in question. All right. So let's look at the different options here for the parasympathetic nervous system. So the cranial nerves are the first options, right? So there's cranial nerve 3, which is our ocular motor nerve going to the glands of the eyes. All right. Cranial nerve 7, or it's the cranial nerve 7, which is here. I'm trying to find which one is it. Cranial nerve 7, which is going to our salivary glands at the facial nerve. Cranial nerve 9 being our glossopharyngeal nerve, also going to some of our salivary glands as well. Cranial nerve 10 being the vagus nerve. If you forget all of your cranial nerves, vagus is the one to remember. It's the most important one. It's the longest of the cranial nerves, and it does the most, right? It's going to innervate your lungs, your heart, your stomach, all the way through to the proximal part of your small intestines, right? So the vagus nerve is basically going all the way through to your the upper part of your gastrointestinal tract. And so that is also going to be the 4th cranial nerve that is involved in the parasympathetic communication. So cranial nerve 3, 7, 9, and 10 will all leave the brainstem, travel a long distance out to ganglia that are closer to their target or effector organs. And then from there, there will be a short postganglionic neuron to the specific organ or gland in question. And then the other option is the spinal nerves, the sacral spinal nerves, which give rise to pelvic nerves. Okay, so these are not somatic nerves. Very important to make that distinction. It's pretty easy to confuse spinal nerves with somatic spinal nerves. So these are going to be pelvic nerves, which are not somatic. So somatic will be things that we can voluntarily control. Instead, these will go to our pelvic organs and glands, like the lower parts of the gastrointestinal tract. So this is going to pick up where the vagus left off, and give innervation to the lower parts of the gastrointestinal tract, so the sigmoid colon, the rectum, upper parts of the anal canal, the bladder, and then the genitalia. So the spinal nerves giving rise to S2, S3, S4, which become the pelvic nerves, will then go all the way out and innervate the pelvic organs and glands. Okay. Now, when we think about the autonomic nerves, these are going to be mixed nerves. As we can see, these have fibers that are going in both directions. So while we will talk about the general route of communication, understand that in a single spinal nerve, there are fibers that are moving to the nervous system, to the central nervous system, right? These are efferent fibers. Oh, sorry, these are afferent fibers. Afferent fibers are picking up information from receptors, picking up information from sensors and detectors, about the world around us, and all that sensory information is being transmitted on afferent fibers with an A to the central nervous system. And then from there, responses and effects and reactions are being communicated on efferent fibers, which will include the autonomic nervous system. So within that spinal nerve, there are fibers that are traveling in both directions. Some are going into the spinal cord, and those will go into the dorsal horn. Right, let me just jump back here to illustrate where that is. Right, so here's our spinal cord. Here's the dorsal horn, or the dorsal part of the spinal cord. Here's the ventral horn. And so sensory information is coming in this direction into the spinal cord. Motor information and autonomic information are traveling out on the ventral. And so in this spinal nerve, there are fibers actually traveling in both directions. So when we talk about these spinal nerves, they're actually mixed in their composition. They have fibers that are both taking up information from the world around us, sending that to the central nervous system. Those are afferent fibers. And then fibers that are sending instructions out to the effector organs, whether that be to skeletal muscle or to autonomic glands, and those are going to be on efferent fibers. So we can already begin to see how the autonomic nervous system is crucial to the loops that we spoke about that help to maintain homeostasis.